Hormonal regulation of the male reproductive system. The male reproductive system is intricately regulated by a series of hormones that control various aspects of spermatogenesis, testosterone production, and overall reproductive function. These hormones act through a feedback mechanism involving the hypothalamus, pituitary gland, and gonads, testes. The regulation ensures the proper development of male secondary sexual characteristics, maintenance of fertility, and effective sperm production. The hormonal regulation of the male reproductive system involves the hypothalamic pituitary gonadal (HPG) axis, which coordinates the secretion of hormones from the hypothalamus, anterior pituitary, and testes. Key hormones in male reproductive regulation: gonadotropin-releasing hormone (GnRH), source secreted by the hypothalamus. Function: GnRH stimulates the anterior pituitary gland to secrete two key gonadotropins: luteinizing hormone (LH) and follicle-stimulating hormone (FSH). Regulation: GnRH release is pulsatile and its frequency can vary. High-frequency pulses favor the release of LH, while lower-frequency pulses promote FSH release. Luteinizing hormone (LH). Source: released by the anterior pituitary in response to GnRH. Function: LH stimulates the Leydig cells in the testes to produce testosterone. Testosterone is crucial for the development of male secondary sexual characteristics, maintenance of spermatogenesis, and overall male fertility. Regulation: LH secretion is regulated by feedback from testosterone. High levels of testosterone suppress LH release, maintaining balance. Follicle-stimulating hormone FSH. Source: released by the anterior pituitary in response to GnRH. Function: FSH acts on the Sertoli cells in the seminiferous tubules of the testes. Sertoli cells are critical for the support and nourishment of developing sperm cells, spermatogenesis. FSH also increases the production of androgen-binding protein, ABP, which binds testosterone within the seminiferous tubules, creating a high local concentration of testosterone required for spermatogenesis. Regulation: like LH, FSH release is controlled by negative feedback mechanisms. Inhibin, a hormone produced by Sertoli cells, negatively regulates FSH secretion. Testosterone. Source: produced by the Leydig cells in the interstitial tissue of the testes under the stimulation of LH. Function: primary function: testosterone is responsible for the development of male primary and secondary sexual characteristics, such as the growth of facial and body hair, deepening of the voice, and development of the prostate and seminal vesicles. Role in spermatogenesis. Testosterone plays a central role in initiating and maintaining spermatogenesis. It acts on Sertoli cells to support the development of sperm cells. Feedback regulation. Testosterone exerts a negative feedback effect on both the hypothalamus to reduce GnRH release and the pituitary gland to decrease LH secretion, thus controlling the production of both GnRH and LH. Additional functions: regulates libido and erectile function, increases muscle mass and bone density. Involved in the maintenance of male reproductive tract structures, including the prostate and seminal vesicles. Inhibin. Source: produced by Sertoli cells in response to FSH stimulation. Function: inhibin selectively inhibits the release of FSH from the anterior pituitary gland, helping to fine-tune the regulation of spermatogenesis. Regulation. Inhibin provides negative feedback primarily on FSH secretion, without affecting LH or testosterone levels directly. Activin. Source: produced by Sertoli cells and other tissues. Function: activin acts in the opposite manner to inhibin and stimulates the release of FSH from the pituitary gland. It helps to enhance spermatogenesis when needed. Regulation: the balance between activin and inhibin is critical for maintaining optimal levels of FSH. Prolactin. Source: produced by the anterior pituitary. Function: prolactin is involved in the regulation of reproductive functions, including its potential role in modulating testosterone production. High levels of prolactin, hyperprolactinemia, can suppress gonadotropin release, LH and FSH, leading to reduced testosterone production and sperm production. Regulation. Prolactin is typically not a primary regulator of the male reproductive system, but may influence it under specific conditions, particularly when its levels are abnormally high. Mechanisms of hormonal regulation. First, hypothalamic pituitary gonadal HPG axis GnRH release. The hypothalamus releases GnRH in a pulsatile fashion. The frequency and amplitude of GnRH pulses determine the relative secretion of LH and FSH from the anterior pituitary. These pulses can vary, with high-frequency pulses promoting luteinizing hormone secretion and low-frequency pulses favoring follicle-stimulating hormone release. LH and testosterone. LH acts on Leydig cells in the testes, stimulating the production of testosterone. Testosterone then facilitates the maturation of sperm in the seminiferous tubules and the development of male secondary sexual characteristics. FSH and Sertoli cells. FSH binds to receptors on Sertoli cells, supporting the process of spermatogenesis by promoting the production of androgen binding protein, ABP, which keeps testosterone levels high in the seminiferous tubules. ABP facilitates the development of sperm cells from spermatogonia to mature sperm. Second, negative feedback loop. Testosterone feedback. High levels of testosterone provide negative feedback to both the hypothalamus and pituitary gland, inhibiting further release of GnRH and LH, thus ensuring testosterone production remains within a physiologically optimal range. Inhibin feedback. Sertoli cells produce inhibin, which specifically inhibits the release of FSH from the pituitary. This feedback loop helps regulate the number of sperm being produced, preventing overproduction. Third, positive feedback regulation. FSH and activin. Activin promotes FSH secretion from the pituitary, enhancing spermatogenesis when needed. This positive feedback occurs particularly in response to a decrease in sperm production or to stress and other environmental factors. Key points in the hormonal regulation of the male reproductive system. Testosterone is central to male reproductive health, regulating spermatogenesis and the development of secondary sexual characteristics. The HPG axis controls testosterone and sperm production through a series of feedback mechanisms involving GnRH, LH, FSH, and inhibin. Sertoli cells support spermatogenesis and regulate FSH through inhibin and activin production. Leydig cells produce testosterone in response to LH, playing a vital role in both spermatogenesis and the development of male sexual characteristics. Disruptions in hormonal regulation. Hypogonadism, a condition in which there is insufficient testosterone production. It may be caused by issues with the testis, primary hypogonadism, or with the pituitary or hypothalamus, secondary hypogonadism. This leads to reduced fertility, low libido, and lack of secondary sexual characteristics. Polycystic ovary syndrome, PCOS, in men, a rare condition where there is an overproduction of testosterone leading to symptoms like infertility, acne, and hirsutism, excessive hair growth. Hyperprolactinemia, excess prolactin can inhibit GnRH release, leading to decreased LH and FSH secretion, which subsequently reduces testosterone production and sperm production. Androgen insensitivity syndrome, AIS, a condition in which individuals with XY chromosomes, genetically male, have a defect in the androgen receptor, rendering them unresponsive to testosterone, leading to the development of female external genitalia despite being genetically male. In conclusion, the hormonal regulation of the male reproductive system ensures the proper functioning of spermatogenesis and the development of male sexual characteristics. It involves a complex interplay between the hypothalamus, pituitary, and testes, with feedback mechanisms that maintain a delicate balance in reproductive health. Any disruption in this hormonal regulation can lead to infertility or other reproductive disorders.